Hi, this is Dr. Pola Rosen, and I'm honored to be here at Cold Spring Harbor Labs with Dr. David Micklos, who will be explaining some of his award-winning research and education endeavors here at the labs, one of the most notable being that 150,000 students are being trained in the area of science. So I think that the STEM, focus on STEM, is certainly prevalent here at the Cold Spring Harbor Labs. Thank you for letting us come and visit, David.
You're welcome.
And would you tell us a little bit about some of the education programs that you have won awards for here at Cold Spring Harbor Labs? What are they?
Well, we provide enrichment for about 30,000 students per year who come here to do experiments, or we do experiments in their classes with them. So that's one element of what we do here.
And what are the grades of the students that can come and learn?
The students who come to us or who we visit in their school range from 5th grade up through 12th grade. So it's quite a range.
Now, if a school is interested in getting involved in these programs and they don't have it currently, how do they do that?
Well, they could look on our website and see the things that we offer. All the school visits are arranged by teachers, so the point of contact is a classroom teacher, either at the elementary, middle, or high school level.
And what kinds of projects do the students actually do? What can a middle schooler do in terms of a project? What would they learn?
The middle schoolers really need to learn across the country that there's variation between living things, and that the variation is inherited to a great extent. And there's a common heredity for things, so that there's some key concepts that young people need to learn, and this is pretty much agreed across the United States in syllabuses, say, in the 4th through the 6th grade. So in fact, for the younger kids, that's what we do, is introduce them to variability between living things and between human beings, and how that relates to inheritance, genetic programs and instructions that a kid or an organism inherits. So those are some key things that happen at the lower level. What we call classical genetics, which is how traits are passed from one generation to the next.
And what about on the high school level?
At the high school level, again, there's a broad agreement across the United States that kids have to learn a few key concepts. Again, they have to do with inheritance, but now the hereditary mechanism, how exactly are genes passed from parents to offspring, and particularly across the United States, curricula have an emphasis on biotechnology, whatever that means to various people, but clearly that means using living things to make products and to run businesses and do new things in the world. Now that tends to be taught in a paper and pen and textbook sort of way, and what we try to do is provide experiments that show how the hereditary mechanism works and how biotechnology works. How would you go about putting a new gene into a plant or animal? How conceptually would you find a drought gene and put it into a corn plant to help us deal with global warming, for example? How would you look at some of your genes and determine that you might be at risk for a disease, or that one drug might work better for you than for me? So these are all concepts that can be gotten at by doing experiments.
I couldn't help but note, as I was making my way to your office, 2 classrooms. The students were absorbed in what they were doing. And in one of them, they were learning about how the firefly lights up. And the other one, they were talking about, or they were working on, the transference of a genetic sequence, I believe it was a DNA or an RNA, in the worm. Mm-hmm. And they were actually doing this. And so the poor worms, were suffering through these operative procedures. But can you tell us a little bit about those 2 projects that we saw here today?
Yeah, so those are 2 good examples of genetic engineering, or things that bear on biotechnology industry. It's pretty dramatic if you take a gene that makes something glow, such as the firefly gene or the genes that you can find in deep sea animals that help them see one another in the dark. Well, you can take those genes and you can insert them into, say, a bacterium, and the bacterium will then take on that property of glowing.
Right, the
Like the
bioluminescence, right?
Yes, like the firefly, or like, the eyelid of a fish that has to attract a mate, at 4,000 feet underneath the ocean. So that's a pretty classic notion in gene manipulation, which is to find a gene that has a particular property or use, and then to put that gene somewhere else where it might be useful for us. That's that experiment that the younger kids were doing. The older kids were working in a field called RNA interference, which is a field that's only a dozen years old, and which received a Nobel Prize just 5 or 6 years ago. So it's extremely new and current field. And that's a field where you can manipulate genes by inserting a piece of RNA. So a little different concept, but it operates at a much higher level in organisms, and it's a mechanism that works better and less obtrusively in higher organisms, such as humans and plants and animals, and in that case, worms.
What do you think Charles Darwin might have said about all of this gene manipulation?
Well, Darwin was interested in Darwin was actually interested also in barnyard plants and animals, and he, among other people, had the idea that if you crossbred two different plants, two different, say, types of corn plant, or two different breeds of dog, that when you did that crossing, that you would come up with a more vigorous organism.
Is that true?
That is true. That's the concept called hybrid vigor, which was in fact rigorously shown here at Cold Spring Harbor in 1908 in corn. But so Darwin was acutely aware that just in the same way that species evolve, domestic plants and animals evolve at the hands of human beings. And he was interested in that, although he didn't know the mechanism, because at that time he didn't know the mechanism of genetics either. Mm-hmm. So, yeah, I think Darwin would say that the things that humans do with plants and animals to bend them to their uses are just human-directed evolution. I don't think he'd have any problem. I think he'd be quite happy with it all.
Well, I'm going to take your word for it since we can't reach him. I wanted to know about online learning. Can you give us some information about what the lab does in online learning?
The DNA Learning Center publishes a lot of websites. We always lose track, but it's somewhere around 28 different websites. The classic thing that people think about a website is something that you learn from, probably by looking at animations and videos and things like that. Mm-hmm. So sort of a multimedia learning experience. And we have a number of websites, probably a dozen or so, that do that, offer you an online learning experience, help you learn concepts especially, but content.
So teachers can tap into this as well as students?
Anyone can.
Anyone,
yes.
And parents?
Yes,
okay.
And so, many people feel that this is an effective way to allow people to learn in a more engaging way than, say, a book, which doesn't have animations and videos. And that's fine, and we have a number of interesting ones in a number of fields, from cancer to neuroscience to the history of genetics to the history of eugenics, which was a not so pleasant period of eugenics in our background. So we have all of that. But I think the things that I'm actually more interested in now are providing tools that allow students and teachers to explore biology in the same way that researchers do. For example, tools that allow you to work with DNA sequence and understand how to get the information from that, or how an organism uses that information. So I'm more interested in tools in a broad sense.
Okay.
And one of our biggest projects which we were just talking about, is called the iPlant Collaborative. And that's a project to provide infrastructure for people to learn about plants and animals, and to provide the infrastructure at a base level that allows researchers to work. But what we're then trying to do is to take that infrastructure and allow all kinds of biology faculty and students to work with that same infrastructure to solve problems. So we have at this point in time a very unique situation in biology education, if you want to talk about STEM education. Because of the large amounts of data that are being generated about genomics and phenomics, which is the traits of an organism, and the great computer resources we have, and the capability of communicating on the internet, we now have, for the first time in the history of science, a situation where students and teachers can work with the same data, using the same tools, and at the same time, as very, very high-level researchers. This has never
It's
occurred.
really phenomenal.
It's never occurred in the history of science that you could do that, but that capability is upon us. And that's my biggest interest right now, is to facilitate that, in fact, students and teachers can work right alongside researchers with the same tools and same data.
Right. Well, David, this was a delight, and thank you so much for your inspirational work. I think for me, one of the most compelling things that you do is to provide hands-on experiences for students in schools of all age groups. And I know that you also send teams out to schools, and there are 2 schools in New York City, Trinity and Spence, that are the recipients of the faculty here and the intelligence. Are there other schools that can tap into the expertise?
Well, the 2 schools we actually work with are Trinity and Chapin.
Chapin, okay.
Although Spence would be a great one as well. Those arrangements are done at the level of the headmasters
Okay.
and curriculum specialists. So,
Okay.
and we're building a very small group of those schools. So we call those schools charter members, because they're charter members of our effort to move into New York City in a big way.
Right.
So we currently have we currently have 2 charter members, and we're looking for maybe a total of 5. So it's quite a select group.
Well, I can only say thank you, not only for speaking with Education Update today, but for influencing the next generation of leaders in science. Thank you so much.
Thank you.